Welcome to ICU Primary Prepcast. Hi, my name is Swapnil, and today we are going to discuss another primary snippet about body water regulation. Now, before we dive into the regulation of body water, for those who wanted to access the show notes of this primary snippet and the previous primary snippets, please go to website www.critcareedu.com.au, and in the podcast series Primary Prepcast, you can access all the show notes of all previous episodes. So, the regulation of body water is governed by high gain feedback mechanism involving the hypothalamus, the neurohypophysis, and the kidneys. When you talk about the body water balance, usually the two key components, one is the regulation of water intake and the second is regulation of water output. Now, the key factors that determine the water intake is primarily osmolality. It is defined as the ratio of solutes in the solution to a volume of solvent in a solution. The physiological threshold for osmolality is 290 to 295 milliosmol per kilogram. Now, if there is an increased osmolality more than 295, the blood becomes more concentrated, the thirst response is stimulated. Now, the primary sensor for this response or this reflex is osmoreceptors in hypothalamus, and that stimulates the conscious awareness of thirst. Also in hypothalamus, there is a supraoptic nucleus and paraventricular nucleus, which secrete ADH. And hypothalamus also stimulates sympathetic nervous system that leads to stimulation of salivary gland secretion. And the result of this overall reflex is increase in water intake. Now, when we have a decreased water intake, that leads to decreased blood volume. Whenever there's a decrease in blood volume, it leads to baroreceptor stimulation and also activation of renin-angiotensin system. The baroreceptor stimulation in turn leads to increased heart rate and increased strength of contraction. The renin-angiotensin system stimulation leads to angiotensin II production, which also stimulates thirst, and aldosterone production, which leads to increased sodium reabsorption from the distal tubule and leads to water retention. And we will talk about these two systems in depth in later half of this primary snippet. So that's all about how the water intake is regulated. So the key two mechanisms, alteration in blood volume and thirst via stimulation of osmoreceptors in hypothalamus. Now, regulation of water output is usually maintained by kidneys. Daily average urine output is equal to approximately 1.5 liters per day. And minimal daily urine production to maintain normal function is roughly around 500 mils per day. In the event of ingestion of too much of fluid, the process of diuresis triggers, usually 30 minutes after the intake, and it reaches the peak within first hour, and it reaches back to normal urine production within 3 hours. Now, there are key systems that play a role in regulation of water output. The first is antidiuretic hormone or arginine vasopressin. The way this hormone works is whenever there's an increase in osmolality, it is sensed by hypothalamus that in turn stimulates the posterior pituitary in supraoptic nucleus and paraventricular nuclei, leads to secretion of arginine vasopressin, which is neuropeptide. Now, AVP in turn results in two actions. One is arteriolar constriction in the peripheral circulation, so that increases the SVR. And the renal effect, it acts via V1/V2 receptors and aquaporin channels, and also vasopressin regulates some of the urea channels. Now, the V2 receptors are important to understand, and I guess in exam you're expected to write in depth about the action of ADH via V2 receptors. The site of these receptors include thick ascending limb of loop of Henle, distal converter tubule, and collecting duct. The nature of V2 receptor is they are G protein coupled receptors, and which results in activation of adenyl cyclase, which in turn leads to increased intracellular cyclic AMP. Now, they are normally located in the principal cell of collecting duct, and on the basolateral membrane side, the AVP or vasopressin acts on the V2 receptor, which in turn leads to increased production of cyclic AMP that leads to activation of protein kinase, which acts inside the nucleus and leads to activation of transcription factors, which leads to increase in aquaporin 2 transcription. And subsequently, there is increased aquaporin 2 channels on luminal side, which leads to increased water reabsorption. In the presence of vasopressin, water enters the principal cell from the luminal side via aquaporin 2 and exits into interstitium through aquaporin 3 and aquaporin 4. So that's how ABP helps to reabsorb more water and leads to antidiuretic action. Now, it also acts via V1 receptor, particularly V1A receptors, which are present in the vasculature of renal medulla. This receptor mediates effects of vasopressin on renal blood flow. This is also G protein coupled receptor GQ11. It activates phospholipid C, thus in turn stimulates the calcium mobilization, which results in vasoconstriction. And V1 receptors are usually widely distributed in the body in addition to renal medulla. The second system that plays important role in controlling the water output is renin-angiotensin system. And again, in this answer, you're expected to write more in detail about this role of renin-angiotensin system in controlling the water balance. So renin-angiotensin system is primarily regulated by renal blood flow. The first stage of this response is renin release from the juxtaglomerular apparatus in response to three stimuli. First is decreased sodium delivery to distal convoluted tubule detected by macula densa. The second stimulus is decreased perfusion pressure in kidney detected by baroreceptors located in afferent arterioles. And the third one is the sympathetic stimulation of juxtaglomerular apparatus. Once the renin is released, it leads to production of angiotensin II from angiotensinogen in liver, and then angiotensin I with the help of angiotensin converting enzyme, which is located in pulmonary and renal endothelium, converts angiotensin I to angiotensin II. Now, angiotensin binds with AT1 and AT2 receptors. Now, these angiotensin receptors are also G protein coupled receptors. However, the most of the actions of angiotensin are mediated via AT1 receptors. Now, the effects of angiotensin, particularly on CVS, it leads to vasoconstriction of arterioles, and the mechanism of action is acts via GQ protein coupled receptors, that's AT1, leads to activation of phospholipid C, that leads to increased intracellular calcium. This in turn leads to increased SVR and thus increased blood pressure. Angiotensin also exerts some of the neural effects. The angiotensin II acts at the level of hypothalamus and it stimulates the thirst sensation. This leads to increased fluid consumption and helps to restore the blood volume by increasing the blood pressure. It also increases the secretion of antidiuretic hormone from the posterior pituitary, which leads to production of concentrated urine and reabsorption of or retention of water. The third mechanism of action, the way the angiotensin contributes to regulation of blood volume is via stimulation of sympathetic nervous system, which releases the noradrenaline and leads to vasoconstriction in order to increase the blood pressure and renal blood flow. Now, angiotensin has also got renal effects. And if you kind of divide them into what is the target, the action, and mechanism. So it acts on the renal artery and afferent arterioles. The action is vasoconstriction, and the mechanism is it acts via receptors, which leads to opening of voltage-rated calcium channels, leads to increase in influx of calcium ions, and thus results into vasoconstriction. Angiotensin also acts on efferent arterioles via activation of AT1 receptors, and then leads to vasoconstriction. And the last action is it acts also on mesangial cells via activation of GQ coupled protein receptors, which open the voltage-rated calcium channels, and this leads to contraction of mesangial cells, which in turn leads to decrease in filtration area, thus allowing the decreased production of urine. It also has effect on proximal convoluted tubule, where it increases the sodium H plus antiporter activity, thus resulting into increase in sodium reabsorption. Now, coming back to the third system which helps in regulation of water output is the role of aldosterone. Aldosterone is a mineralocorticoid. It is a steroid hormone released by zona glomerulosa of adrenal cortex. It also acts at the principal cells of collecting duct, similar to vasopressin. This leads to increased expression of apical epithelial sodium channel or ENaC, and also it increases the activity of basolateral sodium K plus ATPase. This in turn leads to reabsorption of sodium with exchange of potassium into renal tubule. And because it's retaining the sodium, it leads to retention of water along with it. So I guess when you are answering the water regulation, you can either write as a sensor, controller, and effector format, or you can write in terms of regulation of water intake and regulation of water output. Key things to summarize is the role of osmolality and hypothalamus in regulation of thirst and water intake, and also the effect how the blood volume is regulated, which will have impact on the water intake. In terms of water output regulation, is key to mention about the contribution of kidney in terms of role of antidiuretic hormone, role of renin-angiotensin system, and role of aldosterone. So that's the end of today's podcast. We'll be back in week's time with another primary snippet. Till then, goodbye and have a nice time.